Hello everybody, welcome to my poster presentation. My name is Paul Skillen, and today I'm going to talk to you about identifying genes correlated with enhanced CBGA levels in cannabis sativa. That's my master's thesis project, and the poster I'm going to be giving to you today is a little bit different from normal because the setting that we're in is digital, so I've zoomed us all into individual pieces of the poster so that you can see as we're going over the poster bit by bit what I'm actually talking about instead of having to strain your eyes trying to see what it is I'm referring to on a giant poster that's been shrunk down to a tiny screen. So with that said, let's get started. So the demand for various cannabinoid products produced by cannabis sativa has increased in recent years as legal restrictions have fallen away. One of these cannabinoids, cannabidiolic acid, or CBGA, is a precursor to the well-known product tetrahydrocannabinolic acid, THCA, as well as cannabidiolic acid, CBDA. The potential for this cannabinoid to be used in medicine has increased interest at the consumer level, thereby creating a market demand that cannabis sativa breeders can develop cultivars to meet. To assist breeders to this end, we did a transcriptome analysis on a sibling population of relatively high CBGA to THCA ratio plants to identify genes that contribute to high levels of CBGA relative to THCA, which can then be used as a screening tool for breeders. And if you're confused a little bit about what CBGA or CBDA is, I've circled them right here in this biosynthesis pathway. The chemical of interest for my project is cannabidiolic acid, which you can see right here. And the different products that it produces are these three products. And in a recent paper that I'll just point out as one of the potential medical benefits is this one right here, which was published, widely circulated in January of this year. This is something I heard on the radio, and I heard people talking about it to me personally. People wanted to know about how CBGA and CBDA could potentially prevent them from getting infected from COVID-19. And that was something that was really big. It's very topical, of course. And that's one of these potential applications that breeding these plants with higher levels of CBGA could be used for. So the objectives of this particular project are, first, to perform a chemical analysis to verify relative cannabinoid content from a segregating sibling population of cannabis sativa, and I'll explain that in just a little bit. Then next, to perform a transcriptome analysis on the segregating population to identify varying levels of mRNA. And lastly, to identify candidate genes which correlate with a higher production of CBGA relative to THCA, because of course the THCA right now is currently federally regulated, and so we need to make sure that to be federally legal, we want to have high level of CBGA specifically relative to THCA. So for my materials, cannabis sativa cultivar the white right here was crossed with TS5 right here. Then the white is a high THCA content cultivar, and TS5 is a high CBGA content cultivar. This was done to create something that was called W103. Now, W103 for my project was selfed, and it created a segregating population, everything from this relatively high level of CBGA to THCA ratio type of plant to relatively low CBGA to THCA level type plant, and a total of 20 plants, as you can see here, 4 by 5, so 20 plants. Now, each of these siblings was then initially cloned multiple times, and for this project, I selected the 5 clones of each plant, so represented by these 5 leaves, that was most vigorous for the use in this experiment. One of these populations accidentally ended up with a 6th clone, but it leaves a total of 101 individual plants in total. Alright, now moving on to the methods. We started off with flower collection, which was collection of the phytomers, including inflorescences, reduced leaves, and bracts that were all harvested. Any foliage leaves were removed. The plants were then chemotyped, which was identifying the cannabinoid content in each of them. The cannabinoid composition was measured using an Agilent 1260 Infinity 2 HPLC high performance liquid chromatography system. If you'd like to know more about that and how you would do that, I've provided a QR code here to a YouTube video that I made of exactly how you would go about that. Then move on to the RNA extraction, which was done using a Kaigen rna-seq plant mini kit. And there was an additional step that I used for DNA's cleanup steps, so removing any DNA that might have gotten through the filtration. Then the RNA was sequenced using Illumina's NextSeq 2000 platform, 2x100 base pair P3 run for 96 samples for an estimated coverage depth of 41.5, and so 96, you'll notice, is less than the 101 that I started off with. We ended up taking the middle sample in terms of how much cannabinoid content there was, what the middle ratio was, and getting rid of that plant so that we could get a fairly good coverage depth. The fewer samples meant a higher coverage depth. Then the data analysis. So moving on to this little chart that I have here. CBDRX was used as a reference annotation. CBDRX is a low THC, high CBD producing cultivar, which is well researched. Differential expression was analyzed using HiSat2 and StringTie. Specifically, HiSat2 maps the reads to CBDRX reference annotation. Then StringTie assembles the RNA-seq alignments into potential transcripts. The E and B options are used to estimate the transcript abundances and generate read coverage tables. Median normalization was used to transform the coverage tables, and then mean values for each of the clonal population's cannabinoid content and normalized read counts were used to determine genetic marginal effects. Now, all that together, using a correlation test, we were able to produce a set of differentially expressed genes, and that's about where I've gotten so far. Next, I need to be able to look into what the pathways that are most enriched in my analysis to determine what genes or genomic regions are of most interest. But I'll get into that in a little bit here as well. These box plots demonstrate the segregating nature of the levels of CBGA in the population, and that is the distribution is not random, but actually related to the clonal population. You'll see it's not like 6 has some values that are down here below, like around 100, ratio of CBGA to THC of around 100, and some that are up near 250. But they're all clustered in the same region, and that's pretty much the case for all of these different clones. They're all clustered around a certain range of values, and you can see the segregation ranges from close to 100, a ratio of around 100, to as much as almost 300. So we have a pretty good differentiation. You can see that segregation happening throughout this cell population. And now show you the results from the RNA-seq. So these are, I know you can't probably tell what each of these individual values are at the bottom, but each of those represents each of these bars, which is a single clone. And so you can see the clone's values for their coverage depth range from around 25 at the low end to well over 100, which this is suspicious, of course, as it is so much higher than all the other values, so we're keeping an eye on that in particular. But you see the calculated coverage depth was around 41.5, and we got values that were fairly good within what we would expect to have seen, and even outperforming for most part what the calculated coverage depth would be. And then those two things that I just showed you kind of come together. The RNA-seq data along with the cannabinoid content data came together to produce this scatter plot, which shows the positive correlation, as you can see right here, positive correlation between the normalized gene counts for each plant, which is what is on this x-axis right here, and the CBGA to THC ratios for the gene, this specific gene, XM030638089.1, which is the gene that showed the strongest correlation, the lowest p-value. It can be seen here that the plants tend to group by clonal populations. Maybe if you can tell that the 4s are all in this area here, the 12s are all in this region here, the 9s are all together over here. Let's see, what else can we see here? The 5s and the 6s. So this is what you would expect, just to see that plants that are of the same clonal population are giving you the same approximate normal counts as well as the same CBGA THC ratios, and so that's very positive indication that we're moving in the right direction with this. So next steps. This project is not complete yet, but it is currently ongoing. What I'm doing right now is looking at the biological purposes for the highest matching genes. So I just showed you one of the highest matching genes results for the highest correlation values. For several of the highest matching genes, I want to look at the biological purposes of those genes and see, is there some trend? Do we see like a set of genes that are consistently a certain kind of genes that may be the reason why we're seeing such high levels of CBGA relative to THCA. Next, I'll have to rerun this entire pipeline that you see here using the W103 as the reference annotation. Before, we didn't have access to the entire reference annotation. It hadn't been, specifically W103 hadn't been annotated yet, but it has now been annotated by my lab, Brian Knaus at the Kelly Vining lab. And so I'll be using that instead of CBDRX, and that will give better results hopefully than what we had with CBDRX, which was not an exact match to the plant that we're trying to match all of our reads to. And then verifying the results at the end of all of this will be the last step, using qPCR to make sure that the results that we got are actually indeed valid. And with that, I'd like to say my acknowledgments. Thank you to everybody at Oregon CBD for their help in growing and taking care of these plants and funding this project. Thank you everybody at the Vining Lab for their assistance with the extraction procedure in particular. Giovanna was really great at that and showed me how to move forward with that. And thank you to everybody on my committee who volunteered their time and expertise towards this project. And of course, thank you for your time for watching me just now. Oh, and a postscript. If you'd like to download a copy of my poster, I have made it available through, I think, OneDrive. This QR code will take you there directly, or else you can type this code in, this web address into your browser, and that'll take you to the same place, and you should be able to download the kind of a slightly smaller version of the poster that should still be legible. And of course, if you have any questions at all, you can contact me, and my contact information should be available through the app for this program. With that, I'd like to say thank you so much.